All right, we're about to jump into a group of molecules that are, well, pretty much everywhere. They're in the food you eat, they're in your body, they're fundamental to life itself. We're talking about lipids, and I promise you their family is way bigger and a whole lot weirder than you probably think.
So, let me start with a quick question for you. Think about fats, oils, cholesterol, even vitamins like D and E. They seem like a totally random bunch, don't they? But they all belong to this one giant biological family. So what's the one thing, the one property that ties them all together? Well, it all comes down to how they act around water. The official definition of a lipid isn't about what it looks like, it's about its behavior. Basically, a lipid is anything from a biological source that dissolves in organic stuff, but really doesn't like to dissolve in water. You know when you make a salad dressing and the oil and vinegar separate? That separation? That's the classic lipid move, right there.
Now, this whole not liking water thing comes in two main flavors. Some lipids are totally hydrophobic, they're water-fearing, and want nothing to do with it. But others have a kind of split personality. We call them amphipathic. One end of the molecule is cool with water, but the other end isn't. And that little feature is absolutely crucial for building stuff like our cell membranes.
Okay, so with all these different kinds of lipids out there, how in the world do scientists even start to organize them? It turns out they begin with one simple, fundamental chemical test. This is the first and most important split in the entire lipid family tree.
And that big split is based on a process called saponification. It sounds complicated, but just think soap. Saponifiable lipids are the ones that react with a strong base and break down into smaller pieces. In fact, that's literally how soap is made. On the flip side, you have the non-saponifiable lipids. They don't react that way. You can't break them down like that. They're much more stable.
And check this out. This visual makes it perfectly clear. See the reaction up top? That's a saponifiable lipid, a triacylglyceride, getting broken apart. But now look at the molecules on the bottom. Cholesterol, vitamin D, vitamin E. Those are non-saponifiable. You could hit them with that same strong base and they just shrug it off, staying completely intact.
So, let's go down that first branch of the family tree, the saponifiable lipids. These are the ones basically designed to be broken down.
And inside this group, you've got three main players. You have the triglycerides, you know these, they're the fats our bodies use for long-term energy storage. Then you have glycerophospholipids and sphingolipids. These guys are the absolute workhorses when it comes to building our cell membranes.
So how do we tell these different types apart? It all comes down to their core structure, what you could call their backbone. See the molecule on the left? It's built on a glycerol backbone, that part in red. The ones on the right, they're built on something totally different, a sphingosine backbone, shown there in blue. Just changing that foundation completely changes what the lipid does. Okay, let's hop over to the other side of the family now, the non-saponifiables. These are the lipids that are built to last and often have really unique, specialized jobs.
And this family has some real celebrities in it. You've got cholesterol, which is essential for keeping our cell membranes working right and is the precursor for a bunch of hormones. And then you have vital things like vitamin D and vitamin E. See, unlike the fats we just saw, these aren't about storing energy. They're specialists, each with a very specific job to do in the body.
So what do all these different looking molecules have in common? Well, believe it or not, they're all built from the same tiny 5-carbon unit called isoprene. Think of it like a molecular Lego brick. Nature just snaps these little isoprene units together in different patterns to create this incredible range of stable, highly functional lipids.
So we have this two-part system, saponifiable and non-saponifiable. Simple enough, right? Well, that's just scratching the surface. The true scale of the lipid world is just staggering. And modern science has a way more detailed map.
Today, the official system breaks everything down into 8 major categories. You can see some names we've already met, like Glycerophospholipids lipids and Sphingolipids, but it also adds new ones, like Sterol Lipids, that's where cholesterol fits in, and Fatty Acyls, which are kind of the fundamental building blocks for lots of other lipids. This system lets scientists classify any lipid with amazing precision.
And why do we need such a detailed map? Oh, because of the sheer numbers. Just look at this table. It's showing the number of unique lipid structures we found in each of those categories. Take a look at fatty acyls and glycerophospholipids. We're talking about more than 10,000 known structures in each of those categories alone. It's wild.
And when you add it all up, you get this, over 47,000. That's how many unique lipid structures have been identified so far. We're not just talking about fat anymore. We are talking about a massive, incredibly complex universe of molecules, and every single one could have a unique role to play.
So this isn't just about chemists neatly organizing their collections. This whole classification system is one of the most powerful tools we have for understanding biology. Why? Because in biology, structure dictates function.
And that is the key takeaway. This lipid family tree isn't just for organization, it's a predictive map. If a scientist discovers a brand new lipid, they can figure out which category it belongs to, and from there, make a really good guess about what it actually does inside a living thing.
It's a beautiful logical process. First, you identify its main class. Can you make soap out of it or not? Then you look closer at its backbone and its structure to place it in one of those 8 categories. And from there, bam, you can understand its most likely job, whether that's storing energy, building a wall, or sending a message.
And that, right there, is the most exciting part. We found over 47,000 of these things, but we definitely don't know what they all do. We've mapped out the main continents of this molecular world, but there are still so many unexplored branches on this tree. So the real question is, as we keep exploring, what incredible new biological secrets are we going to find?